From the lab to your ears, you're listening to WonderLabs.
Hey everyone, and welcome to WonderLabs. This is Chris Richardson, checking in from the Okinawa Institute of Science and Technology, OIST. I'm really sorry about the buzzing bees in your ears, but I promise it's for a good reason. Because today, I'm speaking with Dr. Maeva Techer. Dr. Techer started out studying honeybees on Reunion Island, an island in the Indian Ocean between Madagascar and Mauritius. But she's now based out here in Okinawa, where she works on the Varroa mite. The Varroa mite is a parasite with origins in Asia that now plagues honeybee colonies around the globe. You might have heard about it for its role in colony collapse disorder, a phenomenon leading to economic loss and a lack of pollination, impacting, among other things, global food supplies. Dr. Techer uses genome sequencing to understand how Varroa has been able to jump between hosts and spread so successfully. And by understanding the Varroa honeybee system, it's possible that she could help to prevent the spread of this parasite, with knock-on implications for colony collapse. In this episode, we talk about the Varroa mite, its origins, the nasty viruses it transmits to harmless honeybees, and its impact around the world. And, of course, the resistance that might give honeybees a chance to survive. We talk about what life is like out in the field, getting friendly Italian honeybees drunk and high on sugar, and some unexpected friends you might never have known as pollinators. We also talk about an exciting new project that Dr. Techer is working on alongside some expert Japanese beekeepers, bizarrely, using honeybees to help save coral reefs as part of the United Nations Sustainable Development Goals. I'll say no more. Without further ado, please enjoy this conversation about our fuzzy little friends and their arch enemies with Dr. Maeva Techer. Maeva Techer, thank you very much for joining us on WonderLabs today.
Thank you for having me.
Let's start in the beginning. What got you interested in bees and mites?
I guess I got into bees first when I was in my master's degree, and I started an internship studying bees in my own island, so Reunion Island. I was super scared, and then we had an amazing professor who was bringing me an opening hive, and I started getting super excited about it. And then, I guess the more I was learning about bees, the more I got very interested. And before actually coming in Okinawa, I didn't see any mite because Reunion was Varroa-free before 2017.
Just to be clear as well, this is a small island close to Madagascar?
Exactly. So it's a French overseas department, which is located between Madagascar and Mauritius Islands. So it's in the Mascarene archipelago. And it's a bit bigger than Okinawa, where we are right now, with a hotspot volcano. And it's often compared as Hawaii, actually.
So there were no mites there, just bees?
Yeah, actually, there was no mites before 2017, and unfortunately, like most part of the world, the Varroa mites that are parasite of honeybee, they actually arrived into the island, and since then, the damage are very terrible for honeybee population.
So let's dive straight into the Varroa then. Maybe you can give us a graphic description of what that experience is like for a honeybee who's been attacked by one of these mites.
Yes. So actually, Varroa mites are ectoparasitic that needs to be on honeybees to survive. And they basically will feed on the fat bodies of the bees, whether they can be adult or larvae. And by that process, then they will survive and thrive into a colony, reproduce, and keep going on. And so one of the things that most people don't know is Varroa was not originally on all honeybees that we know. So the honeybees that most people know is the Western honeybee also called Apis mellifera was the scientific name. And this bee was originally present only in Africa, Middle East, and Europe, and then has been transported a bit everywhere. But the original host of the Varroa mite is the Asian honeybee, which is Apis cerana, and that is originally present in Asia. And so before, like, I would say the 19th century, Varroa mite was present only on its original host, and because these two species have co-evolved for a very, very long time, the damage on the original host was very low. But once this mite was able to jump and do what we call the host switch onto our beloved Apis mellifera, well, the problem is that the Western honeybee didn't co-evolve with it and didn't had good defenses, and since then, like, you have maybe heard that there is colony collapse around the world.
Mm-hmm.
And so one of the main driver of this colony collapse is actually parasitic mites like Varroa mite.
So it's the mite itself that attacks these honeybees in their larval or adult forms, originally in Asia, but it's now spread much more widely. So they bring with them lots of different diseases?
Yeah, so in addition to feeding onto the bees, this mite is also a vector, and so it will transmit a pool of different viruses. And one of the most famous, unfortunately for bees, is the deformed wing viruses.
I don't think that needs much of an explanation for what it does, but yeah.
Exactly. I guess you understand. So when it's transmitted to an adult, it's going to be not too much symptomatic. But if the mite is going to transmit that during the larval development of a bee, well, it can unfortunately make the bee unable to fly and then forage and pollinate.
And I guess this jumping event that took place between the Asian honeybee and the European honeybee, what was the environment that allowed such a jumping event to take place?
So the Western honeybee was present in Europe and was in Africa and Middle East and was kind of protected by a geographical barrier from all the pests that are existing on Cerana. But we came with globalization and beekeeping, and some bees from Europe were actually transported into Russia, where you have Cerana and other part of Asia. And it has been reported after hundreds of years of cohabitation, this artificial kind of sympathy, so a cohabitation between the Western honeybee and the Asian honeybee, gave the opportunity of the mite to basically spill over and test this new host. So we don't know how many trials this might have been doing, but what we know is that there is at least 2 events for the species Varroa destructor in Asia that occurred. And it was reported the first time in 1957, something like this. And then since that, the invasion by this mite, because it has been brought back into different environment, has been very, very fast. And in less than 80 years, this mite has been almost invade globally. And the only part today that remained Varroa-free, and thanks to a lot of effort from biosecurity, is Australia, and also a few islands that are very isolated from continental part.
How do you think about this particular example as an example or a microcosm of global invasions more generally?
I think this is one of the most successful examples of global invasion. And it happened very fast. And the problem is not only on bees, it also threatens somehow pollination services, because we know that bees are one of the most abundant pollinator, especially in crop fields. But the problem also, it's not only the mite is causing damages, but it also brings with it viruses, bacteria, and by the fact that the bees will be more sensitive and damaged by this mite action, it can also make them more vulnerable to other threats that usually they could be able to resist.
So it sounds like a pretty nasty parasite instead of viruses for these honeybees that are actually quite important all across the globe, economically and as pollinators. How big of a problem is this?
Well, I guess as you can hear it from its name, this mite species has been named Varroa destructor because it's a destructive mite. And so nowadays, if a colony is not resistant, so when I talk colony is an ensemble of bees because they live as a social insect, when a colony that is not resistant is going to be experiencing high level of infestation by mite, the colony can die in 2 to 3 years. If you treat that colony, then the colony might thrive, but it means that for the beekeepers and for everything like pollinated related, it's going to imply a lot of cost because you need to treat this colony with a parasite like about every 3 months or every 6 months. You have to get a new management technique. You have to be very careful of feeding the bees when they're getting low or maybe selecting them for better traits. So it's a lot of cost, not only for beekeepers, for bees, of course, but also for pollination.
And presumably this is also having a disproportionate effect in parts of the developing world as well.
Actually, yes. In Madagascar, if I remember, the mite arrived in 2010, and there was, so I don't want to say non-right number, but I think 60% of the colony that were observed after the arrival of Varroa either swarmed or disappeared. And this has caused huge losses for the lychees crop pollination because then the lychee were either pollinated by wind or by bees. And so the yield of lychee produce really dropped that year.
And I do love some good lychees, but I guess
Me too.
that kind of brings it along from the sort of economic consequences for people to global food supplies as well. I mean, as you kind of pointed out already, the honeybees, in terms of their pollination role, that actually might have a more drastic consequence, not just for the individual beekeepers, but also for societal food supplies.
I think it may have a very huge effect, especially because we know that about 1/3 of your food is pollinated by bees. Of course, often we talk about bees as Apis mellifera because it's the most obvious and the most abundant. Luckily, we still have other bees that are still providing services, but the problem is nowadays the most used insect for pollination is still Apis mellifera.
So your research specifically in this area, population genetics of the Varroa mite. Tell us a bit more about that.
Well, I used to work on honeybees for my PhD degree, and I was trying to understand how bees colonize island in the Southwest Indian Ocean and what was the impact of human bringing new colonies, for example, from Europe, because some bees are more productive than other. So you have about 30 subspecies of honeybees. And one of the most favored one is the Italian bee because it's like making a lot of honey and it's super docile.
Making big hand gestures as it does things.
Yes. So this Italian bee is super productive and very docile, and so people will like it and will bring it into environment where they are not present originally. And so what I liked is starting using DNA to be like a detective and understand what happened and what you cannot see. So how those populations evolve and how they colonize this island. And then I came and I visited Okinawa. And that was my first encounter actually with a live Varroa mite. And I was offered the opportunity to study this mite. And I got very excited about it because one of the main issue about this mite is we start to make fight against it, trying to make new control, maybe breed even bees to have specific resistant trait. But one of the issue is we didn't know that much on the mite and the studies were very limited. And one big example of how much we didn't know was that actually until 2000, Varroa destructor was considered as one same species as its sister species, Jacobsoni. And it's thanks to looking at the morphology and the genetic background that we were able to differentiate this species. And one of the main issue is that because of the life cycle and reproduction of Varroa, which is very incestuous actually, so
please elaborate on that.
So what will happen is you will have a mated mother that will come into a cell while a bee is developing. And then she will lay like 3 days after her first son. And then subsequently, like every 30 hours, a sister. And what happened in this closed area is like sisters and brother will be like mating like the Lannisters in Game of Thrones. And so it's kind of bringing, this very high inbreeding. So one of the issues with that is like such inbreeding and the fact that this mite is basically almost considered quasi-clonal means that you have very low diversity. Like especially the one that jumped onto the Western honeybee, they were considered for a very long time as quasi-clonal because only 2 lineage were able to jump. So the very invasive Korean one and one was from Japan. And so one of the issues is that the standard markers that we would use like microsatellite or mitochondrial DNA will not be bringing enough information to be able to study this population. So we had a bit of an obstacle for several years that basically make us study more the bees rather than understanding what's happening with the parasite. And then that's where I come in, and I use next-gen sequencing, and I sequence the whole genome of these mites. So we develop new genomes, and we are using these genomes to understand how this mite basically evolved after the host switch, but also how it spread. And we try to retrace the entire demographic history of this mite after the invasion.
And how successful was that using these advanced DNA sequencing technologies?
Well, I would say so far very successful, since we have been developing 2 reference genome from Varroa. So we have Varroa destructor that I talked to you, but we have also its sister species, Varroa jacobsoni, which is an additional threat and has also switched hosts like in 2008 in Papua New Guinea. So we decided to compare this genome and understand if these mites have evolved the same way. And thanks to this reference genome, now we can basically sequence more additional individuals and try to study the variation that is inside those genomes. So far, pretty happy. And we hope that these new tools that we're developing and new method will be like applicable for everyone who wants to use it worldwide.
So that's a set of quite sophisticated techniques within a laboratory setting to really understand these different sister species of mite and then potentially save the honeybees from this Varroa destructor and
Jacobsoni.
And Jacobsonite. Outside of the lab, though, you also participate in field work working with bees and mites. What does that look like?
Well, we need to go on the field to collect those mites. And one of the big points of this project as well is I cannot go everywhere. And as I told you, it's a global invasion. And we are super happy that we have collaborators all over the world who have agreed to help us in this mission to retrace the biogeography. And they provided us mites from everywhere, so we can basically understand what's happening after invasion. And of course, because I needed to give them some advice, I needed to go on the field and learn more about this mite and know how to collect it the best way, how it is exactly surviving in colonies, basically making the naturalist view of understanding how this mite works. So I can have a better understanding as well when I will analyze the data.
So then you actually have to go out there with a proper outfit on to protect you from bees?
Yes, I guess I'm still like a newbie in beekeeping field, so I still wear this very huge protection. But it depends from a colony to another. So here in Okinawa, the Apis mellifera that is present is the Italian one. That is
super relaxed.
Exactly. Yeah. But I come from an environment where you have African honeybees who are a bit more, I would say, aggressive depending on their mood.
And you have some kind of device that you use to kind of pacify them a little bit, right?
Well, we can use smoke, yes. And also we try to not harm them too much when we study mites. So either we will open brood, so where you can find the larvae directly, where the mites are reproducing. But if I needed a lot of mites, for example, for the genome, what we use is this amazing technique using icing sugar.
Icing sugar.
Yes, so icing sugar, the same for cakes. We'll basically put it on bees for like 2 to 3 minutes, like shake them, and this will not harm too much the colony compared to using ethanol, which will kill your bees directly.
So not getting them drunk, just getting them
Yes.
High on sugar.
High on sugar,
Right.
exactly. And then the good thing is the icing sugar will make the mites fall off the workers, and you can collect the mites in the icing sugar.
So a little less technically complex than your next generation DNA sequencing approach.
Exactly.
Yeah.
Which I like as well.
Yeah. And how do the smokers work?
So the smoker is basically just to tame the bees. Like, if you go in a rainy day, you don't want to distract and disturb them too much. So you put just a little bit of smoke. And this is just with some dry leaf you have. I know some beekeepers use tobacco smoke, which I will not want to use, but
Alcohol, sugar, and tobacco, just
yes, I guess these bees, they get high on everything.
throwing them all the addictive things we can find. Yeah. So until now, a lot of your work has focused on the mite itself, and you mentioned earlier about these collaborations you have around the world. What might the other side of the equation from this parasite-honeybee interaction tell us? How might studying the honeybee help us to understand what's going on or potentially develop some solutions?
So yes, luckily we have a lot of researcher teams that are working on understanding potential resistance from honeybees. So we have hope on that side. So what's happening is it's about 80 years now that the mite have been invaded progressively worldwide. And in the beginning, you had a lot of honeybee population that collapses just by the presence of this mite and the viruses that it transmits. But recently, about in the last 10 years, some researchers have found that around the world, there is some natural honeybee population that are surviving to this mite. So one of the things we don't know is it potentially the mite background being different? Is it those mite are particularly different? Or is it the honeybee itself that has been developing this resistance? For example, in Africa, you have two populations in South Africa and Kenya that have developed a suppressed reproduction of the mite. So the mite is not as fertile as you would expect. In Avignon, in France, they also found that the hygienic behavior was much higher than in other places. So it's kind of a lot of hope that these bees are developing this resistance. And I think what researchers are trying nowadays is identifying the genetic mechanism or any other mechanism, evolutionary mechanism that could explain this resistance. And on my side, I'm actually looking at those mites that are from this resistant population and see if eventually this is the same mite you will find in a susceptible population. And if it's the same, then it means that the resistance is really just developed by the bee and it's not somehow an unbalanced equation between this parasite-host interaction.
And if that resistance is coming from the bee, how might that resistance be evolving and how would you go about studying that?
Well, one of the problems is these resistance rates are really great, but I don't think they will be able to completely help remove the mites or completely control it to a level that they're inexistent. And also you have to remember that we are in a host parasite system with insects, which have a very short generation time. So actually the evolution might be very fast for the mite to overcome this new resistance. So it's a constant battle between the bee and the mite. And we hope that this resistance might just make the level of infestation lower, and that long term, maybe the bee itself will be able to evolve so many resistance, just like the original host, to make this damage very low. But for that, we have to go through a natural selection process, I guess, because it will not be possible to just achieve that by management technique. This is my opinion, of course. I hope we can deal with that very quickly, but I doubt it.
So in the meantime, while we wait to see how that resistance pans out, the original host that you mentioned already is very resilient against the mite. And I'm aware that you've got an upcoming project that actually involves using those original hosts to try and untangle what might have led to them being so resilient against the mite. Can you tell us a bit about that?
So one of the amazing thing with this original host, so Apis cerana, it has co-evolved for a very, very long time with those parasitic mites.
And that's the Asian honeybee.
Exactly, the Asian honeybee. And so the original host has developed a few defenses that are very interesting. First, the bees are able to groom themselves, and they can even recruit nestmates to say, hey, please help me to remove these mites off my back. Then they are also able to perceive where larvae might be infected, and so they will be able to uncap the cell, check if the cell is potentially infested, and recap it if it's not, if it's considered healthy. In the case it found that it's infested, it will be able to just remove the mite and even remove the infested larvae to reduce the number of future offspring from this mite.
Yeah, I've seen some videos of that in action, and it looks like a very sophisticated behavior. It almost looks like a conscious bee that's making the snap decision whether or not to eject something from the hive.
Yes, what is amazing is this behavior is not only just for Varroa mite, it's for other disease, because you can have, for example, brood disease that is just making those larvae very smooshy and full of bacteria. So the bees will be able to just remove the infested or the unhealthy larvae. And I think one of the most interesting for me, although I never could see it in action, is called the entombing. So the bees will wait the mite to go into the drone brood, which is apparently more attractive to the mite because the drone will have a longer time of development.
So the drones are not workers, the drones are the
males.
The males that mate with future queens?
The future queens, exactly. So it's only seasonal. They will appear only when the right conditions are for the colony because they don't bring anything else than mating with the future queen. And so what is very interesting is these drones will have a much longer time of development. And so the mites are also very attracted and they can even co-infest, which means that those bees, those Asian bees, will set traps, leaving the drone being completely infested and just recover it with wax and let them die. So basically the mites and the infested bee will be entombed.
Expendable males. Yeah.
Exactly. I think it's quite amazing, those different resistance. And so one of the thing is some of the Western honeybee have started to develop kind of similar resistance, like the Varroa sensitive eye gene, which is the way that these mellifera are able to detect a parasitized cell and just remove the mite, or also like this higher grooming behavior. But one of the thing we find very interesting is that with all this panel of defense, Serana is able to make the level of Varroa infestation so low that it's almost undetectable for us when we go on the field. So we wanted to understand if part of those mechanisms were not only behavioral, because, for example, one of the things that is really amazing, it has been reported in different places that Apis cerana, so the Asian honeybee, is able to completely suppress the mite reproduction. Meaning that a mother mite will enter a cell, will be having all the right condition to be able to lay eggs, and yet when the bee will hatch, no eggs will be produced at all. So one of the reasons that could happen is like you have this kind of invisible biological warfare between the bee host and the mite. And in our team, we are actually people with so many different backgrounds. And my colleague Vienna Kovalik, who's a microbiologist, had a very nice discussion with me where we were thinking that maybe microbiome might be some help in this biological warfare.
So that's very similar to the microbiome that we have in our own guts.
Exactly. So I believe that you have some kind of core microbe that are present in those bees, and they are very important for their survival. And we don't know if there is additional one that could help them fight pathogens. And what we would like to do is align basically my population genetics background with her microbiology background using a comparative study by looking at the original host and the Western host both placed in the same environment and seeing how these bees will react to the same mite, for example.
And how is that project shaping up so far?
Well, we tried a first time. Unfortunately, the Okinawan weather was not really happy with us.
Blame it on the weather. Yeah.
Exactly. Because we had some Japanese honeybees, so Apis cerana japonica, who's also presenting this very high resistance trait against Varroa destructor. And we brought 2 colonies like a year ago here. And unfortunately, typhoon wiped them out. And then we had been contacted by Ona Village office, who was actually starting a nice project.
And this is the village just up from the campus.
Exactly. Not so far. We are actually living in Ona Village. And so this project was for them to have more bees, and they wanted to use bees for a particular project about red soil. And we find this as a good opportunity to actually collaborate with them and their expertise as Japanese beekeepers, to work with us with those Japanese bees and the Western honeybees.
So from my understanding, red soil, the thing that they're choosing to focus on, it's this big problem in Okinawa where partly as a result of agricultural farming, you have these monoculture crops. So that when it does rain and those typhoons that you've been describing, or just regular rains, as opposed to being more thoroughly absorbed by the soil, these agricultural crops tend to change the soil in such a way that the water kind of saturates on top of the surface, and then it gathers up some of that soil, that red soil sediment, and it forms these small streams and rivers that kind of run off into the ocean. And on certain days, you can actually see the ocean turning this red color as well, because there's just so much sediment, and that can have really catastrophic consequences for the coral reef ecosystems around the island. So part of their task is to try and minimize the amount of red soil that is reaching the oceans. So in order to do that, it's not like you can just stop agricultural farming on an island that relies on that for a part of its economy, and obviously the individual farmers who need that money as their main source of income, and also just the food supply, right? So they approached you for your knowledge on bees because bees might be able to help. Is that fair?
Yes, it's a really weird way to connect those 2 concepts, but I love the idea they had. So what they wanted to have, and I think the simplest sentence is bee saves coral.
Just leave it at that and end the episode. Yeah.
So the idea behind it is just to promote beekeeping into unused land of farmers and encourage them to grow more flowers or more wild plants around it that beekeepers will enjoy putting their bees in front of it, so the bees can produce a lot of honey. And it's kind of a collaboration between beekeepers and farmers. And by that way of promoting more collaboration between these 2 pans of agriculture, they hope that they will help put a lot of plants around Ona, which will be able to reduce this red soil phenomenon. And so since this project is just starting, really kick off this year, what we wanted to do is like bring our own honeybees that we have in our experimental apiary and just help this project by putting some of these bees into pilot farms so the beekeepers can start seeing how this work. And for Ona Village, we will have a special honey, like the bee save coral against red soil honey. So we're really excited about this. And one of the good thing is we have one of the only colony of Japanese honeybee here in Okinawa. So this will be a very special honey for this project.
Yeah, I think it's, as you said, it's a very unusual, very interesting, very counterintuitive approach to tackling this environmental concern. And the overarching picture that fits into is that the local government has been tasked to assist with the United Nations Sustainable Development Goals. So this is all part of some big global collaboration. But I guess what it means right here is that, yeah, this repurposing of agricultural land that's currently just been abandoned or is not being used, and then these other strategic points within agricultural farming plots to help put that to an end. And then at the same time, you and your team are able to perhaps untangle some of the mechanisms underpinning resistance in the Asian honeybee, the Japanese honeybee, and then potentially bring that forward to see how that information could help with colony collapse disorder.
Yeah, exactly. I think it's always nice to have part of an applied opportunity with our research and also more collaboration with locals and general audience. So it also helps us to promote more about what we are doing and explaining to local Okinawan people what are we doing and what are the bees able to do against this parasite that most of the time they don't know of.
Yeah, and also just getting to enjoy some delicious, sticky, artisanal Honor Village honey.
I think this is lovely.
Well, before I let you go, we usually like to wrap up with a quick fire round.
Okay, let's go.
Can you explain in a few simple words your research area?
So I am studying Varroa mite, which are parasites of honeybee population using DNA sequencing and a lot of bioinformatics to understand how these mites have invaded globally the world.
Which emerging field in science or tech are you most excited about?
So I guess because I started with next-gen sequencing and all this data analyzing that you need bioinformatics, I really enjoyed seeing new techniques such as machine learning to use for analyzing this data. So I'm really looking into that into the future.
If you could wave a magic wand and erase a common misconception about your research area from everybody's collective memories, what would that be?
I guess the easiest, because I'm working with evolution, is that evolution is not just a theory. But another way to see that is also that evolution is not as progressive, like in an advantageous way that people think about it. It might basically bring new traits that are not beneficial for the individual or that are just neutral. So it's not always in a good progressive way.
Is there a tool that you use in your work that you're particularly fond of using?
I guess my pipette in the lab.
Why is that?
Oh, I started working with it since my master, and they have been my main help in in the lab to prepare my libraries and sequencing.
Do you have a science-related joke or fact for us?
Not a joke. Actually, we were talking about bees, and most people's conception is that pollination, so the fact you have plant being reproducing thanks to animal, is been done by insect or birds. But one of the most interesting case of pollination for me are on islands, where you have some case of lizards, such as geckos, who are very fond of nectar and will by that way pollinate flowers.
What? So which island was that?
Well, you have that in Mascarene Archipelago, but you have even other case where you have unexpected pollinator, like in South Africa, you have some mongoose that are doing pollination just for a candy tree, taking some nectar, and by that process, they're helping flower reproducing.
Where can people go to find out more about your work?
So I guess you can look at our group website, so the Ecology and Evolution group at Okinawan Institute of Science and Technology, and our website is homologo.us.
HOMOLOGO.us.
Exactly.
Maeva Techer, thank you so much.
Thank you, Chris. It was a pleasure.
Thanks for tuning into WonderLabs. If you enjoyed this episode, please take a moment to subscribe, leave a review, and share it with others who you think might enjoy it. You can also email us at wonderlabspod@gmail.com. This is your host, Chris Richardson. Our brand was designed by Lloyd Preston Allen, and our jingle was produced by T. Fitzgerald at For The Record. You can find more music from For The Record at soundcloud.com/WEAREFTR. Special thanks go out to Liliana Laborde-Dozian. We'll see you next time.